we are at the top of the hour. Thank you all for joining. Dr. Quintero, I will turn it over to you.
Absolutely. Well, good evening. This is super exciting. I am Dr. Diana Quintero. I'm the Director of Pediatric CF Care Center at East Tennessee Children's Hospital in Knoxville. And I want to welcome you to the Cystic Fibrosis Education Night, hosted by the Tennessee Consortium of CF Care Centers and produced with the support of Tennessee chapter of Cystic Fibrosis Foundation. The Tennessee Consortium is a collaboration initiative of all cystic fibrosis care centers across the state of Tennessee, and that includes Children's Hospital at Erlanger in Chattanooga, Children's Hospital at Vanderbilt in Nashville, East Tennessee Children's Hospital in Knoxville, Le Bonheur Children's Hospital in Memphis, University of Tennessee Health Science Centers in Memphis, University of Tennessee Medical Center in Knoxville, and Vanderbilt University Medical Center in Nashville. So these are both the adult and the pediatric centers. Our goal is to provide the cystic fibrosis community across Tennessee the best care, and by collaborating on best practices, supporting one another in challenges, and working together, because we know that together we can do this. Speaking of support, I'd like to invite Dr. Somini Srinivasan, the director of the Pediatric CF Care Center at Le Bonheur, to come and give some thank yous to those who have supported this event tonight.
Thank you for the introduction, Dr. Quintero. Tonight's event has been a wonderful collaboration across many facets of the cystic fibrosis community of care. First and foremost, I would like to thank the nurse coordinators of the CF Care Centers across the state of Tennessee for your leadership in planning the content and structure of this first ever virtual and statewide CF Education Night. This was quite an undertaking in a very new format, and your hard work has not been unnoticed. So thank you for this, and thank you for all you do for our patients every day. I would like to say a very special thank you to the Tennessee Cystic Fibrosis Foundation chapters. Thank you, Lee, Megan, and Maddie, for all your hard work in making this event happen. I would also like to take a moment to recognize the sponsors of this evening's event: AbbVie, the makers of Creon; Chiesi, the makers of Bethkis, Perdsey, and Bronchitol; Genentech, the makers of Pulmozyme; Gilead, makers of Caiston; Kroger Specialty Pharmacy; Nestle Health Science, the makers of Zenpep; Vertex, the makers of Kalydeco, Orkambi, Symdeco, and Trikafta; Viatris, the makers of Tobipod Haler and Hillrom. Many of the reps from these pharmaceutical companies are online with us tonight, and please join us in the chat box to say thank you, and please feel free to directly message them with any questions or comments you may have about their products, as they are a wealth of information and insight. Now, I would like to invite Dr. Rebecca Brown to come on and introduce our plenary speaker for the evening. Thank you.
Thank you. It is so nice to see everybody joining this event. It's nice to bring everybody together virtually when we can't be together in person. I'm Dr. Rebecca Brown. I'm the director of the CF Center at Vanderbilt, and I'm thrilled to introduce our plenary speaker for the evening, Dr. J.P. Clancy. Dr. Clancy is a pediatric pulmonologist and the Vice President of Clinical Research for the Cystic Fibrosis Foundation. Previously, he served as the Division Chief of Pediatric Pulmonology at the University of Alabama at Birmingham from 2003 to 2010, and the Director of Research for the Pulmonary Medicine Division at Cincinnati Children's Hospital Medical Center from 2011 to 2018. His role at the CF Foundation is to oversee clinical research conducted by the Foundation's Therapeutics Development Network, as well as investigators from academic institutions internationally. He also is deeply involved in the education and training of physician scientists and career development in CF research. Tonight, Dr. Clancy will be sharing with us what's new in CF research, from better ways to treat symptoms of CF to even better CFTR modulators in the exciting world of genetic therapies. Dr. Clancy will give us an overview of what is currently under development and a look into the future of what could come in CF research. Please join me in welcoming Dr. J.P. Clancy to our virtual stage.
Thank you so much for that kind introduction. Good to see you, Rebecca. And can people hear me okay? Give me a thumbs up. Yes? Okay, good. Just wanted to make sure. Really a pleasure to be with you tonight, and particularly being with Tennessee. I mean, I think of all the things that I love about Tennessee. I lived in Alabama for a long time and just live a little bit further north now. Still have family down in Alabama. Get down there quite a bit. You know, start with the ribs in Memphis, and then you kind of move your way over to Nashville for the hot chicken, and then you go over to Knoxville and you're in the Smokies, and then you go over to Chattanooga and you're at Lookout Mountain. So anyway, love them all, and really a thrill to be here. I'm going to go ahead and share what I hope is the right one. Here we go. And hopefully you're seeing something that says what's new in CF research, and assuming that you are, then we'll go ahead and get started. And I'm just going to make sure this is going to do what it's supposed to. So just a quick disclosure, again, the CF Foundation does invest in companies for early development of CF drugs, and if those actually come to market, sometimes we are actually able to recoup some of those investments. Certainly can learn more about it on our CFF website, but all of our funds that we raise are reinvested back into CF research and care. So I wanted to give really today just 4 general areas, a very quick overview of CF. I'm quite confident that many in this audience are quite familiar, but in case there's people who are newer to CF, maybe don't hear the background as much, happy to just briefly talk about what that is. Then talk a little bit about symptomatic therapies that are under development, and those are treating the symptoms that result from the loss of CFTR, the gene responsible for CF. Then we'll talk a little bit about sort of recent developments with CFTR modulators, and finally, what's kind of on our horizon, genetic therapies, and what lays in front of us. And again, very exciting days, I think, are really in store for all of us. So a reminder that although we often talk about the lungs and the digestive system in CF, actually it is a full body disease. You can find symptoms of CF in a lot of different places. A number of different organs are shown here. The common theme here is that in CF, there is a gene that doesn't work correctly, and its name is the Cystic Fibrosis Transmembrane Conductance Regulator, or CFTR. And I won't say that whole name anymore tonight, just I'm going to keep it short because it is definitely a mouthful. And when it doesn't work correctly, that's how the symptoms of CF occur. It is a genetic disease. It's found in about 1 in 3,000 births in the US. It varies depending on what country you're in, somewhat higher, some have a bit lower rate. There's around 33,000 people in the US and over 80,000 worldwide, probably an underestimate because it's probably present in a lot of countries where it's not really looked for very carefully. And as noted here, a number of organs are involved, and the CFTR gene can be broken in up to, it's been described as many as 1,700 different ways, but the common theme for all of those is that the mutations lead to loss of working CFTR. And if we look at sort of the steps that lead from that genetic problem to the symptoms of CF, and looking at the lungs, we start with the lack of working CFTR. CFTR is basically the instructions for the cell to make something called a protein, and the CFTR protein, its job is to help move salt and water in different parts of the body. And in the lungs, when that CFTR is not working correctly, mucus tends to be thick and it builds up. It is prone to getting infected. Your body responds to that infection with lots and lots of inflammation, and that causes more mucus and so forth. And over time, the lungs get damaged. And what we can end up with, which is shown here, unfortunately, is someone who's undergone a transplant, looking at their lung, the very damaged airways. And what we see here are the airway tubes themselves are very large. That's what these kind of large open spaces are. And you can see in some of them have thick mucus and pus, basically infected secretions in the lungs. And obviously that is the one thing we clearly want to avoid, and many of the new therapies we think are really changing this dramatically. So if we look over time, there have been a lot of advances in CF since it was first described way back in the 1930s by Dorothy Anderson. She was a pathologist in New York who described a series of cases of kids who had died young, very, very early in life with nutrition problems with malnutrition and also lung infections. And over time, we've been able to add lots and lots of different therapies. And you can see them here: pancreatic enzymes, anti-staph antibiotics, ways of clearing the lungs, anti-pseudomonas infections, Dornase alpha, which is pulmozyme, ibuprofen and azithromycin, which we think help with inflammation, nebulized tobramycin, hypertonic saline, aztreonam or casein. And then we said we can see that through all this time, there's been significant improvements in life expectancy for CF patients. And indeed, that life expectancy now has crossed over 50 years, which is amazing. And you can see on the top here a number of sort of important landmark things, such as sweat testing was identified, development of the CF patient registry by the CFTR gene was identified, adult standard care, newborn screening. And I think what's important is we've now entered this era where these drugs, which actually restore function to dysfunctional CFTR, are becoming more and more available. And with that has really been a transformation in what CF is and what CF can be in the future. But before we go there, I wanted to summarize some different types of research that's happening right now. There's lots more going on, but I can only hit on a few highlights, and certainly happy to take any questions during the Q&A around these topics. But what I have here is just a picture of the CF drug development pipeline. And what we have moving from left to right are sort of different classes of molecules, and the height of each of these different little bars is how far along it is in clinical development in clinical trials. And the purple guys are the CFTR modulators and other things that work to fix CFTR. The green is things that help with mucus. The blue is things that help with inflammation. The orange is things that help with infection. And then over here in the darker blue are nutritional things. And the point is there's lots of different things that are under development. And I'm just going to highlight a few of those things over here on the right side that are might be of relevance and interest to people here. First, I think we have to acknowledge that CF and lung infections have historically been one of the highest concerns, if not the highest concern in the CF population. And because of this, the CF Foundation initiated a $100 million commitment 2.5 years ago called the Infection Research Initiative. And with that, a very broad-based program was developed with the goal of really, advancing, bringing people into this space to try to develop new drugs to treat CF infections, and also help advance them, and also help better understand those infections and how we can manage them. This commitment is almost going to be crossed. Actually, we've covered that 100 million in about 2.5 years, and there'll be an announcement coming probably at the very end of this year about when that actually happens. And this has really sparked a great deal of research in the space, and a number of options are now coming closer to fruition for treating difficult to treat infections. One example that people may have heard about is phage. A phage is it's a virus basically, but this virus attacks very specific bacteria. And there's been a lot of reports in the media over the last few years about people with really difficult to treat infections which have been successfully treated with phage. Now, we don't know what kind of role this would have in CF. Is this something that would be used to treat when people are having increased symptoms, or is it more of a chronic therapy like some of our other inhaled antibiotics? But we have initiated the first study of phage in CF, the first controlled study. The company's name is Armada, and there are 3 additional companies who are also in the midst of developing phages to treat different aspects of CF lung infection. So very excited to see what the results of those are over the coming years. The next one I wanted to highlight is something called STOP2. And this was a large study that enrolled well over 1,000 people, really trying to standardize how we treat pulmonary exacerbations. These are basically periods when the lungs infections become more symptomatic and patients often require IV antibiotics and time in the hospital. And what they found in this study was that for patients who responded quickly to their antibiotics or showing benefits quickly, it didn't matter if they stayed 10 days or 14 days for IV antibiotics. They really had the same outcomes if you looked at it both ways. And another study that's going to be coming behind this called STOP3 is going to be doing some other types of work, looking, for example, at the benefit, whether use of one antibiotic is sufficient, or do we always need to use 2 antibiotics for pulmonary exacerbations. And finally, I mentioned down below STOP-PEDS. This is a study that is more focused on young kids who have increased respiratory symptoms, and really looking at the role of whether to quickly add antibiotics by mouth, or see if increased symptoms, which are usually caused by a virus, can resolve before antibiotics are necessary. And this study is nearing completion. It's just enrolled a little over 100 people and should be completed over the next coming several months. As I mentioned before, inflammation is another problem in CF lungs. And this is basically your body's response to infection. And over time, inflammation, while it controls infection, can damage tissue. And so we have been trying very hard to identify drugs that may help take the edge off some of that inflammation and therefore limit any damage that can happen to the lungs because of the body's response. And I mentioned 3 programs here that are currently in clinical trials, both in the TDN and also in sites outside of the US. One by a company called Laurent is looking at basically a vitamin A derivative that may be able to help treat infections. And that work we are hoping will be reported later this year. The second one is a company is called Calithera. They're basically developing a drug that is trying to restore a natural antibiotic that our body makes called nitric oxide. It's normally something, kind of a gas actually, that is released at low levels in our lungs and helps to treat and keep infections at bay. And people with CF tend to have low levels. And so this approach is trying to restore nitric oxide levels and help to improve the lungs' ability to fight infection. And the third, Santhera, this is a molecule that is geared at blocking an enzyme that we know can damage the lungs. And that enzyme is called neutrophil elastase. We've known about neutrophil elastase for a long time and felt that if we could actually temper the activity of that, it is released by white blood cells, and it's good at killing bacteria, but it's also very good at stirring up more inflammation. And so we're hopeful that that type of a drug will really be able to help reduce inflammation in the CF lungs. I also want to highlight some other areas of focus that don't really have anything to do with the lungs. The CF Foundation has been partnered with the National Institutes of Health for over a decade, really trying to better understand CF liver disease and can we predict those who are at risk, and are there ways that we can prevent it? And this study is called PUSH. It's a group of approximately 12 centers that have been involved doing this and really trying to understand the role of ultrasound screening to determine those at risk for developing liver disease. Only about 5% of people actually go on to develop symptomatic CF liver disease. And so we often have a hard time knowing those who are at higher risk early in the disease. Second idea that is looking forward to getting started hopefully in 2022 is called the CF, looks at treating CF-related diabetes with something called a bionic pancreas. And basically what this is a combination of basically software that ties together continuous glucose monitoring, a very common way for people now to monitor blood sugar. It's usually just a disc that sits on the skin and gives you a readout with your phone. And that message can then be sent to an insulin pump, which is able to automatically manage the blood sugar. And so this study is really another partnership with the NIH, and really may be a great advance for people with CF-related diabetes. And finally, I mentioned the Mayflower study. This grew out of a working group established to really look at women's health, looking at sexual reproductive health and other aspects of CF disease and interplay with women's health. And they have developed a study that has just started enrolling. It's really looking at maternal and fetal outcomes of patients during pregnancy, really carefully looking at those who are and are not being treated with modulators like Trikafta. And so this study is going to be big. It's going to enroll approximately 300 women, and we'll follow the children up to 2 years to confirm that there really are no concerning findings for people who do take modulators during pregnancy. A study of this scope has not been conducted yet, and it's really needed. And finally, I want to mention a bunch of studies that we're under development, and some of these have to do with CF lung disease, and a lot of them don't. One where it's called NICE-CF is really going to be looking at different ways to use colon cancer screening in CF. We know that patients and adults with CF are at a much higher risk of colon cancer, and so finding ways to really use some of the newer testing to sort of identify those who need more frequent scoping would be really valuable. Strong CF is under development, and this is a study that's really looking at the relationship between nutrition and advanced lung disease. We know that people with advanced lung disease often have trouble with nutrition and vice versa. We want to better understand how those two are interrelated. Outreach is a study that we're developing that's focused on examining how home versus clinic spirometry performs. I think we all realize that during the pandemic, there's been a huge shift to the use of telehealth and somewhat swinging back, but for a lot of people, perhaps in the adult community, use of telehealth is likely to continue. And we're trying to better understand how the data that we get from home compares and contrasts with that we get in clinic. We're also going to be looking at fungal infections, and specifically some people who are at risk for fungal infections. How do we really better define that? How can we get specimens in the home rather than always relying on going to clinic? Those studies are getting off the ground this fall. And then finally, I think we've all recognized that there are significant mental health challenges that can come with CF, certainly for the family, certainly for patients and children and young adults and older adults. We know that CF patients have a higher risk of anxiety and depression, and we're working on developing a more sensitive screening tool. And we also want to better understand the full TRICAPTA experience. Clearly there's been benefits, dramatic benefits seen for patients treated with TRICAPTA, but there's also been some other symptoms that have come on, and whether cause and effect is difficult because this has all sort of happened in the context of the pandemic. We certainly hear reports of people having potential questions about mental health and relationships to modulators, and so we certainly want to dig in deeper to that, better understand if there are things that we need to be screening and treating more aggressively. So I'm going to switch gears for the rest of the talk, and I'm going to talk a little bit about therapies that try to restore function to the CFTR protein and really get at the root cause of CF. And as I mentioned before, the gene responsible for CF was identified back in 1989. That guy that you see sitting there, his name is Francis Collins. He's just getting ready to retire from being the head of the NIH for a number of years. But he was actually a CF researcher and was one of the leaders in identifying this gene way back when. But because we have that information, we were able to start to develop therapies that actually with the goal of bringing CFTR function back in people who have CF. And there's different ways that you can do this. As you can see on the far right is the picture that is showing examples of treating the manifestations of CF. In other words, those downstream symptoms that we see when CFTR is missing. But on the left side, we have different approaches. One being trying to restore function to the protein. That's what modulators do. One trying to restore levels of RNA, and that is would make sense for those who don't have any. They have a problem with making the CFTR RNA, and these are called biosynthetic defects. An example of these are people who have stop or nonsense mutations. And finally, DNA strategies, in which we would actually give back a full normal functioning CFTR that either could be beside or theoretically incorporated into the genes of CF patients. And this is what gene editing and gene delivery is. And so we'll talk a little bit briefly about those. So modulators, again, are basically, we call these things small molecules. They're basically molecules that have been identified different ways, but usually by sort of identifying, doing screening to find drugs that are able to sort of activate CFTR in a laboratory setting. And this example is showing here an example of a CFTR chloride channel. We know CFTR is a protein. The protein codes for channel. That channel is basically a pathway for salt to get across cell membranes. And when CFTR is not working, it won't open correctly to let the salt through, or sometimes actually it gets sort of stuck inside the cell and can't actually get to the cell membrane where it's supposed to do its job. And what CFTR modulators do is they either help it get in the right shape or help sort of flick the switch that allows that channel to open and be in the place that it's supposed to be so that it can do the job it's supposed to do. And these modulators all in general though require that you have some CFTR protein available for them to work on. And I wanted to just remind people of just the dramatic effect that TRICAPTA has had. It truly is an amazing drug that has had profound effects on the CF community. And I will highlight just some of the key results from the study that was led to its FDA approval back in 2019. What we're looking at here in the top is the lung function of patients in that study after they started TRICAPTA, and you can see an improvement in lung function that was on the order of about 13 to 14% compared with those who were on placebo or basically were not receiving active drug. And hand in hand with that was the reduction in sweat chloride values by more than 40 points below the diagnostic threshold for CF on average. And so this was for patients who had one copy of F508, that being the most common CF causing mutation and the one that this drug seems to work most effectively on. and really was profound and opened the door for people to have access to very potent CFTR modulators across the board. More recent studies have kind of confirmed this in younger age groups. This is looking at patients treated with Trikafta who were ages 6 to 11. This was a tricky study to do because it was actually happening right during the pandemic. So getting data and getting results was really challenging, and kudos to all the research teams who were able to actually help complete this study because that data was sufficient for the FDA to grant approval. And what we see is very similar looking data, maybe even more effective, seeing that these young kids who by and large have very normal lung function have an improvement in lung, very rapid improvement in lung function, FEV1, within 2 weeks. Again, 10% improvement on top of already normal values. And looking over here at the sweat chloride values, again, seeing even arguably a higher drop or a greater drop in sweat chloride with values actually dropping more than 60 points. So really down even below this, getting back even closer to the normal value. So the kind of the upper end of normal. So really very, very exciting work that was conducted and completed throughout the pandemic. So the CF Foundation is trying really hard to understand CF that is treated with Trikafta. And I have the term here, ETI. I hope that's not confusing. That's basically an abbreviation for the 3 drugs within Trikafta, elexacaftor, tezacaftor, and ivacaftor. Those are ET and I. And we have a number of studies, and I'll show you some high-level results that really are just fresh off the press from the PROMISE study. And the PROMISE study is really looking at patients who initiated Trikafta as part of care. In other words, they've been prescribed this drug and we are monitoring a number of things in much more of a real world type of setting. These are patients who may or may not have even qualified to be in the original study. They maybe have been too healthy or too ill or somewhere in between. Point is, though, this really gives us a sense of how this drug might work in sort of the real world once we get out of the confines of a clinical study. And I highlight some other ones here, which we won't go into great detail on, but we're going to be enrolling almost a quarter of the entire CF community in the United States in CF clinical research to better understand ETI. So I hope that your research teams are at some point will be having conversations with you about some of these studies and really is helping us better to understand what CF is in this new world with Trikafta and other highly effective modulators. And what I was just going to show here is some improvement for different groups of people in different measurements. And the far left here, we're looking at lung function. In the middle, we're looking at sweat chloride. And the right, we're looking at a quality of life instrument called the CFQR. It's basically a questionnaire. And starting over here on the left, we're looking at lung function improvement. We're looking at people who are starting on, because Trikafta is available for people who just have one copy of F508, it's sort of created a number of different groups who might actually either start or switch over to Trikafta. So the folks in blue are those who are on no modulator at all because they were candidates before. The green, who are those who are on something like Orkambi or Symdeko. And then the orange are those who are on ivacaftor. And we know that that's the group that seem to have the most effective modulator, although the numbers of patients who qualify for ivacaftor is kind of small. And what we saw was very similar results compared to the clinical trial. Those who had been on no modulator had a huge jump in lung function. And what was kind of, to me, really interesting, and even those who are on ivacaftor, a drug that we know is really effective for a small number of mutations, had actually a pretty good improvement in lung function. So telling us we can do even better than what we used to think we could do before. And moving into the middle here, looking at sweat chloride values, again, those who either started modulators for the first time or took switched from Symdeko or Orkambi, which are kind of modestly effective, had a big improvement that we could see here. And even those who were already on ivacaftor had another drop in sweat chloride of 25 points. So really their value was actually much lower, just the relative change is smaller since they started at a lower value. And then finally, symptoms. We considered improvement in symptoms of 5 points on this scale to be important or significant. You can see huge jumps of 3- and 4-fold above what we consider normal, so or significant. So I think the key point is once this drug started to be used in clinical care, we continue to see these benefits across a much wider range of patients. I think another thing that's important to see is that by and large, people the majority of people who start ivacaftor were actually able to stay on it. So these are looking at the different visits in the study, the number of people who were completed the visit, and the number who were staying on Trikafta. And we can see this visit 3 is a lower number. That was right in the middle of the pandemic. But for example here, for 487 patients who started here, at 6 months, we had 98% of patients were still able to stay on drug. And so we don't know yet about those who did not complete, were not able to stay on drug. We do know that some patients have problems with liver tests, some people have problems with rash, there are other symptoms that other people have, but we definitely want to better understand that so that we can ensure that people who are candidates for modulators can get the benefits. And I think the other point that this study wanted to make is that even over 6 months, something like sweat chloride remains really powerful marker for that the drug is active. So what we think CFTR modulators will at least ultimately allow us to do is to treat, approximately 93% of people with CF who have either one copy of F508, at least one copy, and or one of the other much less common mutations that have been shown in the laboratory to respond to Trikafta. But there is clearly going to be a group of people who will not be good candidates, people who have CF caused by mutations that don't make any protein, and often these include nonsense mutations and other different types of mutations, which unfortunately just are not going to work going to be a good option for modulators alone. So I'm going to spend the last probably 10 minutes just talking about where we're going in terms of genetic therapies. And, I think of this as back to the future. Actually, when I got started in CF research, it was back in 1989 when the CF gene was identified. And soon after that, there was a lot of work in gene therapy in CF. And it wasn't successful. And I want to be able to send the message today that we're actually in a very different place. And I'm really excited about what the future holds for genetic therapies in CF. So if we do look back, there were about 18 clinical trials that were conducted mostly in the 90s, a few into the 2000s. And most of these were looking at ways of sort of putting the DNA that coded for CFTR into cells for a transient period of time, for basically the life of the cell, if that makes sense. These tended to be small studies. They usually were just at one center. Industry was really not particularly involved. There were a couple of companies that were involved, Genzyme briefly, another company called Targeted Genetics, but a lot of the stuff was actually being done by academic laboratories. And common themes, the main things we found is that there were 3 general approaches that people took. One was basically what's called a non-viral vector. It was basically DNA plus a lipid carrier. These just were not very efficient and could also be inflammatory. A cold virus that had been weakened to carry these genes had been used. It tended to cause some inflammation, and it also caused an immune reaction so that it was very difficult to redose people. And then finally, a virus that doesn't make one particularly sick. It tends to be a very, very sort of mild, almost trivial type of infection, but it is known to be able to carry CF genes, but it just wasn't very efficient. And we were not very good at making this. So long story short, unfortunately, things were not looking terribly rosy for gene therapy back in the 2000s, up into 2010. So why should we be enthusiastic now? What's different about today that wasn't the case back then? First of all, CFTR modulators have told us that CFTR makes sense. If you restore CFTR function back to people with CF, you get lots and lots of downstream benefits. And I know that sounds obvious, but actually back when we started this, we didn't know that. And knowing that CFTR makes sense as a way to treat CF is definitely helps us keep it in the bullseye. Second, there has been an explosion of research and advances in genetic therapies in many, and not for lung diseases, but for other diseases. Diseases like sickle cell, spinal muscular atrophy, thalassemia, which is a blood disease, certain eye diseases that are, again, typically are sort of rare genetic disorders. But there are literally hundreds of active clinical trials going on right now in examining gene therapy. And so we've learned so much more than that we had when this was last tried. We have a much better toolbox, meaning we understand CF much better than we did. We have better coordinated efforts across different research labs and also industry labs. We have better model systems, in other words, ways to study giving genes to cells and mRNA to cells. HBEs or even bronchial cells. These are basically cells that we can grow in the laboratory that really are a good model and really were the workhorse for getting CFTR modulators into CF patients. Better animal disease models and new and better vectors. When I mentioned some of these approaches on the far right, new approaches, these didn't even exist when we were doing gene therapy before. The idea of giving mRNA as a therapy just wasn't there. And we've clearly learned through the pandemic that mRNA-based vaccines are very good at developing a protein. For COVID-19, you make a protein that your immune system recognizes and tells it to kill the virus. Well, what we would be using it for is to use your cells to actually make a working CFTR, not something that your immune system recognizes. But the point is, it has moved so quickly with this. Stem cells. We didn't have any idea what the stem cells were in the lungs. These are the cells that are the progenitors. They give rise to all the cells in the lung, and they help keep repopulating the cells. And finally, gene editing, the idea that you can go with molecular scissors and actually fix someone's gene. That concept didn't exist until about 2013. So huge changes have happened. I'm not going to lie, there are still significant barriers and challenges. We know that when we look at the CF airway, it's got some unique challenges. The mucus is thick and it often has lots of debris in it, like infection, white blood cells, and so forth. And furthermore, the lining cells of the airway, they spend all day trying to clear that mucus out with cilia, which are these basically little finger-like microscopic projections, which just beat all day long to help clear the mucus out. And those very good at helping clear out some of these kind of ways of putting the genes in. Other, sort of things that the airway cells do, their job is to keep things out, and they have a very tight connection between the cells, which is not easy to penetrate. And I guess the point of this thing is it's clearly it's not going to be a slam dunk, super easy thing to do. But the point is, I think we're in a much better place than we were just even 5 years ago. And we understand the cells that we think we need to get to. I know this is just a cartoon looking at airway cells, but used to be we kind of thought about these cells more or less, a lot of them were the same or sort of mixed. But we've learned actually that different cells have very different functions in the airway cells. We have the ciliated cells, which again beat the mucus all day. We have secretory cells, which help release that mucus and also keep it well hydrated. We have basal cells, which are the ones that actually sort of provide, or sort of the source cells for the other ones. And then finally, these new cells called ionocytes, which really have lots and lots of CFTR and seem to be really important in regulating how that whole epithelium works together. I wanted to just highlight some programs that are sort of either getting close or actually maybe in clinical trials this year. And I say this because this is really at our doorstep. We believe that I'm quite confident that we will be doing genetic therapy research with medications delivered to the lungs of CF patients in 2022. We've already had protocols submitted to the TDN, which is one of the network that helps to sort of organize and conduct a lot of these clinical trials. And they're coming in different flavors. One is using, as I mentioned before, mRNA and lipid. There are 3 companies right now that are in this space. One is Translate Bio. It's actually already had reported phase 1 study results. 2 other companies are not far behind, Arcturus and Recode, and believe that they'll be in clinical trials within the next 12 to 18 months. Therapies for nonsense mediated CF. There's one already in clinical trials. The company is called ELOX, and they are currently in a phase 2 study for patients who have the G542X CF causing mutation. A couple of other companies are screening new medications that we believe may be able to help restore function to these different types of CF causing mutations. Icogen is one, and another is called SRI. And Recode also has a program that's built upon a novel approach using a different RNA molecule. We're also supporting a company that is really doing sort of an interesting approach called Splysense, which is really for a small number of mutations, but really is very sort of specific in that it helps restore the normal processing to certain types of CF causing mutations. And so the first one that they'll be studying is listed here, and look forward to some of those results coming soon from that work. And in addition to those other programs, we have a number of gene transfer studies that are ongoing. In fact, the one company that I mentioned here, 4D Molecular, has already submitted a protocol for us to review in plans of getting into clinical trials in 2022. Two additional companies, one called Spirovant, another called Crystal Biotech, are also developing viral-based vectors, which are really a sort of using modified viruses to carry the gene of interest in. And these are really sort of exciting and really groundbreaking approaches that they'll be taking, which were not available only 5 years ago. And gene editing, I would say at this point, this is again is conceptually where you go and actually fix someone's gene. This is definitely happening in the laboratory, and I think we're still, at least for CF, a few steps away from this happening immediately. There's some clear barriers to overcome, but the exciting thing is that this type of approach, if successful, would be able to theoretically restore CFTR function to all the cells and maintain the normal regulation of the gene like it's supposed to, because it's already just sitting in the genes of that person. And again, the goals of this, when we talk about gene editing, is a permanent fix. We would certainly be thinking about starting with the lungs, but we would hope over time that we would be able to target these to go to those cells where CFTR function contributes to signs and symptoms of the disease. So the CF Foundation really has jumped in boldly into this space. They've committed over $500 million to try to stir interest by industry to come into the space and also to start to answer some of these fundamental questions. And I won't go into great detail here, but just noting that we're trying to create new science by supporting laboratories. We're trying to support what we call infrastructure, or sort of tools that allow us to compare and contrast how different approaches work, investing in new companies and new technologies, taking on new approaches rather than simply just having contracts with companies, actually trying to build companies and partnering with venture capital companies to build CF-specific companies. And then finally, a number, just highlighting down below that up to 10 companies have already been invited for funding in 2021. And again, this is going to be really active space for the next 5 to 10 years. I think it's also important for us to realize that in terms of clinical trials, we're definitely going to need to adapt to doing genetic therapy research. This is a map of the sites in the US that are CF care centers, and the size of the population of patients who are not eligible for CFTR modulators. So basically, the bigger the circle, the more patients there are who are not modulator eligible because of their genetics. And what we see is it varies a lot. There are some places like out here in California, in Arizona, down here in Houston, you have larger Hispanic populations where there's a much larger number of patients who may have rare mutations that are not candidates for CFTR modulators. Same thing up here in the Northeast, down in Florida, here we go in Atlanta, up here in Minneapolis, Seattle. Point is, we're really going to have to start thinking about how we bring these studies to those patients, and also how do we prepare the CF community and also the CF care and research teams to talk about genetic research, because this is all new territory for most people in this space. So to prepare ourselves, we've really assembled a really tremendous group who are working together to really have ourselves prepared. We have a work group that is focused on preclinical guidance for industry, basically letting our industry partner know what are the things that we would like to see from the laboratory before we bring these studies into human subjects. We have a group focused on educating care and research teams, finding out what the needs are so that they can help to have good conversations with potential CF trial candidates, educating the community, finding out what they do know and what they don't know about these different approaches to genetic therapies. And finally, a work group that is really focused on helping figure out what's the right type of study. How big does it need to be? How long do we need to follow them? What are the things we should measure to know that we're actually impacting the CFTR gene? And then finally, just highlighting that we've tried to do this in the context of our current clinical research framework, rather than creating something new, really try to build it on top of those things that we do currently. So I just, in closing, I want to just highlight a few things. Remember, the CF therapeutic pipeline is very full. There is a lot of activity across a lot of fronts, and I have a lot of enthusiasm for some of the research projects that are either happening now or soon to be entering clinical trials. These include symptomatic therapies, new modulators, and genetic therapies. I didn't even mention that there is a new study of a Vertex triple combination called VX121 which is just getting off the ground, which is going to compare head-to-head with Trikafta to see if it's a benefit. And a cool thing about that new drug is it's only once a day, and that certainly is could be really beneficial for people and adherence to their medicine. We know that clinical trials of new modulators and genetic therapies are really on our doorstep, and we're really trying to set the stage to do those studies efficiently and really effectively, and really being highlighting the importance of partnering with the CF community to be able to be successful. So that is it. And so I hope I stopped sharing, and more than happy to take any questions. But it is, yeah, these are exciting days. You know, there's actually a few unknowns out there. You know, how exactly these genetic therapy studies are, which one's going to be likely to be more successful, kind of to be determined. We're not sure. So anyway, more than happy to take any questions. But thank you again for your for listening in.
Dr. Clancy, I'm Joel Ledbetter. I'm the pediatric director in Chattanooga at our CF Center, and I will be facilitating questions and answers. And I have a few questions, but first, just thank you for being here. Thank you for your excitement. It's very contagious. And seeing your timeline, I began CF career in 1980, and we didn't know much, couldn't do much, and results weren't very good. And look where we are now.
Oh yeah.
And it's just amazing. And certainly we have a lot of families here, and that's due to all the support from families and friends of CF, and it's supporting research and supporting the care centers. And so, you know, just a huge thanks to all of them for doing that. So go ahead.
I was just going to say, none of this happens without you. You know,
no, absolutely.
We can't move anything without partnering. So thank you.
I've had a few questions. Some of them were given to me earlier.
Sure.
And one of them, and I'm asked this a lot also, about any thoughts in the pipeline when Trikafta will be available for the younger children, those under 6.
Yeah, it's a great question. I would imagine that just following you, I don't have any inside information, so I can give you that. So this is, you know, opinion talking, but just sort of knowing how generally how the FDA kind of works on these things. They obviously will need to have a sufficient, what they call it, basically a safety package, and also the sort of good understanding of the dosing for kids under the age of 6. And they are completing studies in patients ages 2 to 5 right now. And and typically those studies usually are observational for about 6 months. So, and then once that's done, then the data is sort of put together, provided to the FDA, and they have several months to review it. So I would say I think it's possible that we could be talking about by the end of 2022. You know, that's you know, don't hold me to it, but I think it's possible. You know, if everything goes right, and obviously there's a lot of things that have to come together, but I don't think that's unrealistic to think that we'll have dosing available for that age group. So very excited, obviously, to hear that. And then obviously we'd love to continue to see these things continue to march down. You know, the younger we think we can start, the potential to prevent problems before they start, potentially rescue pancreas function. Those are all, you know, on the table when we start talking about earlier starting.
Yeah, I have a question from, we have in our Tennessee CF Consortium, we talked about this a lot, and this is the question from Dr. Quintero. Yeah. And it has to do with treatment burdens. How do you envision a change in daily therapies? I get asked this every visit.
I imagine you do. You want the short answer? We don't know, but
yes.
But I mean, if you stop and think about it, none of the therapies that were developed for CF were studied in the context of sort of working CFTR, you know, which is where we are right now. So it doesn't mean we stop, but it certainly does raises a question to say, all right, well, maybe not everything is necessary for every person. And I think that's where we're trying to, you know, I think what we'll end up with is much more sort of person-based approaches. You know, there's going to be like, this person really benefits from, you know, hypertonic saline, and homozyme. Well, that person, you know, maybe who is started modulators at a younger age doesn't really have problems with chronic infection. Maybe those are things they only use when they're having symptoms, you know, and maybe that's where we're going to get to. We are doing one study that is really trying to look at this, at least in a systematic way. The study is called Simplify. It's enrolling a lot of people for a relatively short period of looking to see how does lung function and symptoms do when people basically are stable on Trikafta and they either stop their hypertonic saline or stop their Pulmozyme or stop both. They have sort of different options to look at. And I think the truth is, it's going to vary. I think there's going to be some people where it's clearly beneficial and necessary. I think there's probably going to be people who get such a bang out of the, out of their Trikafta or whatever that modulator may be, that benefit of the other stuff is smaller. And when you survey people, you are finding, yes, there are significant numbers of people who are, taking a little hiatus from this therapy and that. And I think the main thing is we just want to watch carefully. If things are changing, we should know. And I think that's, if you're having those kind of conversations with your docs, just have honest conversations. That's probably the most important thing I can say. We are trying to get at these questions, but it is going to take a little time to get the studies completed. So.
Okay. Another question here. I was asked about rare mutations, and I think you answered a lot of that very well. Here's a very specific question from one of our care centers here about splice mutations. If you had any comments on that.
Yeah, so there's kind of two types of splice mutations. There's ones that are called canonical, and the other ones are called non-canonical, which of course is confusing names, but basically the canonical ones or the non-canonical ones sometimes make a little bit of, they have some of their splicing works and some of it doesn't, is basically. The non-canonicals, unfortunately, they rarely work at all. I'm sorry, the canonical ones, but that one group, I gotta stop saying this, one group, there is some functional CFTR. And in fact, ivacaftor is actually approved for some of these splice mutations that have a little bit of function. However, another approach, which at least in the laboratory it looks kind of promising, is the one I mentioned about SpliceSense, which basically uses something called an oligonucleotide. It's basically a small stretch of DNA, basically, but it basically goes and sort of helps the splicing machinery do it right as opposed to do it wrong. And sort of tips the scale into normal splicing, if that makes sense. And so that approach, though, is very sort of one mutation at a time. That's a little bit of the challenge of that. You can't sort of study in a bunch of, it's very much sort of depends on, it's only going to work for one given mutation at one time. So that's kind of the challenge that comes with that. Now, all of the genetic therapies, mRNA therapies, things like that, actually apply to anybody who's got honestly, they apply to anybody. These replacement therapies, in theory, could benefit people who, don't get a full bang out of their buck, and for whatever reason, the modulator doesn't work well for them, or they have other mutations that don't respond to Trikafta or otherwise. So these genetic therapies, ultimately, we're talking about doing studies in small groups, but they could be applied to well beyond that group. And who knows, maybe we'll be talking about add-on therapy for people who are getting full pulmonary benefit, for example. So,
a question that's come up in our consortium, we talk about, we can't get sputum cultures anymore.
Yeah.
And you were mentioning doing things at home. We have trouble even in clinic.
Yeah. It has become a challenge, and we actually had a workshop literally last week focused on what's called detection and diagnosis. What are the, and we are in this really strange place. Do people who are not able to give us any sputum at all, that's good. That's great. I'm glad they can't cough it up. But is everything okay down there? Is there any bacteria that we're just not seeing? And so that's what we're really kind of wrestling a little bit about. How do we best do that? There's some neat ideas out there, to tell you the truth. One that I just sort of highlight that's been under development. I don't think it's quite ready for prime time. But basically, they do an analysis of breath and look for sort of signatures in what they call volatile organic compounds, or VOCs, that come from things like Pseudomonas or staph or whatnot. And so rather than actually getting a specimen of mucus, you can actually maybe just breathe into this bag and then go have it analyzed. So again, it's a little bit of science fiction, but I've seen it work. It's really cool. And so I think there's some great ideas down there coming if they're not here right now.
Well, I think we're getting near the end, so I'm going to ask one more question, and you can use this to close it if you like. And the question is, with all the new medications in the pipeline, how can people get in to participate in research?
Yes, well, great question. If you are interested, the CF, first of all, you can always talk. I mean, I think you can always talk with your CF care centers about your interest in research. I know a number of the centers here are part of the TDN network. There is also other research opportunities. But if you were trying to find a central resource for, find out if you are a candidate for being in a clinical trial, the CFF, if you go to the CFF website under research, there's something called a clinical trial finder, which basically you type in information and it can tell you about studies that are happening across the nation and where they are happening. And we want you to come and be interested in research. As I mentioned before, nothing changes without you. We have to have the CF community engaged. And that's, as we get into these genetic therapies, this is going to be, we're talking with people, we'll be talking about doing studies in people who haven't done research before. And this is going to be really important. So anyway, please reach out to your care center teams. If you want to do some online stuff, I think the foundation has some great resources. Clinical Trial Finder, it's just all one word. You just type it in there and it'll take you right to it. So it's great.
All right, if you have no further comments or have any questions, I guess that would be it. And thank you very much.
Of course. Hey, it's my pleasure. Thank you for having me, and thank you for being part of the inaugural. Is it the inaugural sort of Tennessee CF Consortium? Is that true? The first joint big meeting? That's awesome.
It is, and we've even added somebody from Mississippi. So
even better. All right.
Even better. Yes.
All right. Go Balls. No, wait a minute. I better be quiet. So
thank you.
Thanks again. Bye now.
Yeah.
Thank you, everybody, for joining our plenary session. We will be taking a 15-minute break, and we will then begin our breakout round 2. So feel free to just keep the link live and open. Hang tight. We'll put back on our slideshow to thank our sponsors one more time for this evening, and enjoy your 15-minute break. Thank you so much, Dr. Clancy, and all of our physicians for facilitating this plenary session. Enjoy your 15 minutes.